Okay, welcome my students. I'm Rukia Abdallah, a biology teacher. Today, I'm going to concentrate myself in the topic of transportation and a subtopic transportation in plants. I'm going to make my presentation by following 4 questions. Let us check question number 1. Question number 1 is this one. It has been written that, why is transpiration considered to be necessary but evil process? Think it. Why is transpiration considered to be necessary but evil process? In order for you to answer such particular question, you are required to consider advantages and the disadvantages of transpiration. And before discussing these advantages and the disadvantages, it is better for you to define the word transpiration. You should first define what transpiration is. For instance, you may say transpiration is the process whereby water is lost in the leaves in the form of water vapor. After defining what transpiration is, then you are required to go directly to advantages of transpiration. Why transpiration is necessary? Why? The question of why transpiration is necessary, it means you concentrate yourself in advantages. For example, you may say that transpiration brings about cooling effects in plants. Cooling effects, or you can say it cools the plant. That is one. Second, transpiration helps mineral salts and water to be conducted from the soil up to the upper part of the plant. Okay, here you can add more advantages of transpiration. Here I just gave you 2 examples of advantages of transpiration. After that, you are going to tackle the second part. That is disadvantages or hazards of transpiration. Why is it a disadvantage? For example, here you can say that transpiration leads to dryness of the cells. It means cells lose more water and it becomes flaccid, and that flaccidity, if it is severe, it can even lead to death of the cells and the plant as a whole. That is one. Second, here we know that plants need water for various purposes. For example, photosynthesis needs water. Even the removal of waste products in the body of the plant need water. Therefore, if water lost, it hinders other physiological processes of the plant due to lack of water. From there, you can add more other disadvantages of transpiration. If you can manage to get either 5 here, disadvantage, and 5 advantages, it means now you can say that you are able to answer such particular question. Okay, after knowing why transpiration is considered to be a necessary evil, we are required ourselves to discuss the following issues. Where does it take place? Where does that process take place? You know that transpiration may take place in 3 places. First, stomata. Second, cuticle. And the last one is lentil cells. Lentil cells. If transpiration occurs through stomata, we call that transpiration as stomatotranspiration. If it occurs in cuticle, we call it cuticular respiration. And if it occurs in lentil cells, we say lenticular transpiration. But mind you that high amount of water loss takes place in stomata rather than these 2. In transpiration here, we have seen the meaning of transpiration. We also discussed the importance of transpiration. We discussed the effects of transpiration. We discussed the sites where transpiration takes place. Now, it is better to differentiate transpiration and another process which is known as guttation. What is guttation? Ask yourself, what is guttation? Okay. In short, we can say that guttation is loss of water in plants in form of liquids. In transpiration, water is lost in form of vapor, but guttation, water is lost in the form of liquid. Therefore, it is your time now to make differentiation, to differentiate or to distinguish between these 2 terms. You can construct the table. You can distinguish using table with the 2 columns. Column 1 for transpiration and the column 2 for guttation. Then you are supposed now to point out your points based on the differences between these 2 processes. For instance, you may say that in transpiration, water is lost in form of vapor. While in guttation, water is lost in form of liquid, and it is pure liquid. Number 2, you may also consider time. At what time transpiration takes place, and at what time guttation takes place. Mind you that here transpiration is commonly during the day. It is common during the day. But guttation is common at night. Apart from that, you may consider also humidity in the atmosphere. I think you know what is humidity. The amount of water vapor in the atmosphere. So, if there is high humidity in the atmosphere, then it is difficulty for transpiration to take place. Because of what? The gradient there will be minimal. Therefore, transpiration will not be favored. But it favors guttation. You may also consider other factors. There are so many factors which govern transpiration and guttation. In order to know those factors, you are required to read. Read your books. You may use biological science as a reference. You may also use understanding biology. You may also use the new book which we have from TAE. They explained each and everything concerning with that. If you use those books, you will get a lot of information about the differences between respiration and guttation. Question 2, describe the structure of a stoma. Stoma, singular, plural, stomata. In order to answer such kind of question, as I said earlier in previous presentations, I said that description can be made using words only, or you can include even diagrams. It depends. So, description there of stomata. We know that it is made up of Golgi cells, 2 Golgi cells. These 2 Golgi cells look like a kidney or bean-like structure. And the inner wall, this wall, is much thicker. And the outer walls, that means these ones, they are thin. Okay, now to answer question number 2 about the structure of stoma, you are required to draw the structure like this one. We know that stoma is made up of 2 Golgi cells. These 2 Golgi cells are epidermal cells. They play part in controlling opening and closing of the stoma. When you check the structure here, you may observe that there is the inner wall, which is much thicker than the outer wall, which is somehow thin. The cells here, these Golgi cells, contain other cell organelles, like chloroplast. Therefore, apart from controlling the closing and opening of the stomata, These cells also synthesize food. Okay, the Golgi cells is surrounded by other epidermal cells. It have been surrounded by other epidermal cells, like the diagram here shows. These are the two Golgi cells, the inner walls, which are much thicker than the outer walls, which are thin. The space here is the stoma. These structures are the other epidermal cells. Apart from that, these other epidermal cells also contains other parts like vacuole. The vacuole is this one. The vacuole is there, and other structures. And after discussing this structure, then you will be in a position to go and discuss another structure. But mind you that the stoma can be opened or closed. When it closes, it looks like that diagram. You see, there is no aperture here. No aperture. The aperture or the stoma have been closed. It means, it shows that the stoma is closed. Mind you that the stoma should open to allow entry of gases, also should close to prevent water loss. And when it opens, it looks like that diagram. You see it. At the middle here, the space is open. That is the stoma. The stoma is open. I think you have understood. After discussing the structure of the stoma, then you are required to discuss the so-called mechanisms of opening and the closing of stomata. I'm going to use the question there. That is question number 3. Question 3 says that use the following hypothesis to explain closing and the opening of stomata. There are so many hypotheses which tried to explain how the stoma opens or closes. But in my presentation here, I'm going to tackle only 2 hypotheses. That is starch-sugar hypothesis and the ionic theory hypothesis. I'm going to start with A, that is starch-sugar hypothesis. Okay, for the case of starch-sugar hypothesis, it says that during the day, in the presence of light, plants synthesize food in Golgi cells. This food, which is synthesized there, makes the solute potential in the Golgi cells to be more negative. Solute potential becomes more negative. But apart from that, the water potential becomes, during the day, plants synthesize food. When food is synthesized in Golgi cells, solute potential there becomes more negative. And when solute potential becomes more negative, water potential becomes low. Why? because of dissolved sugars. When sugar dissolves here, it lowers water potential compared to other epidermal cells. Due to that, osmosis will take place. Water will move from the area of high water potential to the area of low water potential. That means from other epidermal cells to the Golgi cells. When they enter the Golgi cells, the membrane there, the outer walls, expands very fast compared to the inner walls. Because the inner walls, I said that they are thicker than the outer walls. When they expand, the cells becomes turgid. And that turgidity leads to the opening of the pore there. The pore becomes open. That pore is what is called the stoma. The stoma becomes open. But in darkness, vice versa takes place. But you don't write vice versa takes place. You are required to explain. In darkness, Golgi cells do not synthesize food due to lack of light. If food is not synthesized there, we expect now the solute potential to become less negative. Solute potential becomes less negative. At the same time, water potential becomes high. This makes it water now to move from the area of high water potential to the area of low water potential. From Golgi cells to nearby epidermal cells. In so doing, the stomata closed because the Golgi cells becomes flaccid. That is why the stoma closes. That is how the stoma opens and closes based on a starch-sugar hypothesis. But apart from that, I said that I will also make revision with you on ionic theory hypothesis. In answering that part B, I'm going to use the diagram in order to make you understand more. I'm going to start with when the stomata is closed. For instance, here the stomata is closed, and it is closed during darkness. At that time, more potassium ions and chloride ion will be to the outside. When I say outside, I refer to other epidermal cells. But when light is present, it activates the enzymes known as ATPase. The enzyme ATPase will be activated. When this enzyme is activated, it converts ADP to ATP. This ATP, which have been activated here, will be used to pump hydrogen ions. To pump hydrogen ions from Golgi cells to other epidermal cells. When these ions goes to other epidermal cells, they again enter by carrier protein together with another ion, that is chloride ions. When chloride ions enter there, also potassium ion from other epidermal cells enter the Golgi cells. The influx of these ions in Golgi cells makes the Golgi cells to acquire low water potential. Water potential there becomes low due to these ions. The result of that is the entry of water. Water will enter the Golgi cells by osmosis. When water enters, the stoma opens. I think you have understood. Let us now check the diagrams there. Check this diagram. The stoma is closed. The stoma is closed, but when light is present, it activates the ATPase enzymes. Why? It means that ATP activates that enzyme. The enzyme is responsible for the conversion of ATP here. ADP is converted to ATP. This ATP is used to pump out hydrogen ions. When hydrogen ions pumped to the other epidermal cells, then they enter again to the Golgi cells through carrier protein. They carry also ions like chloride ions. Also potassium ions enter the Golgi cells. The influx of these ions allows the entry of water. Let us check another diagram there. You see, water enters the Golgi cells. These Golgi cells become turgid, and you see it. I said that the outer wall is thin. Due to thinness, it makes these layers to be more elastic compared to the inner walls. That is why the Golgi cell bends inward and creating the aperture here, which is the stoma, and at that time we say that the stoma is open. I think you have get something from there. Up to there, we are through with that question. We go to question number 4. Explain mechanisms of transport of manufactured food materials. Explain mechanisms of transport of manufactured food materials. Okay, in order to answer such kind of question, first of all, you are required to know that food materials is transported by phloem. Food is carried from leaves and all other chlorophyllous cells. When I say chlorophyll cells, I think you understand. The cells with the chlorophyll. Because photosynthesis takes place in those cells with the green pigments, particularly leaves. After that, we expect food which has been synthesized by leaves to be transported to different parts of the plant. Through phloem. Different parts here includes roots and all other parts. And leaves here, which synthesize food, are considered to be source. The source synthesizes food, that is the leaves. After being synthesized, the food is carried by phloem and it is poured to the sink. Those parts which use food are the ones which are considered to be what? Sink. For example, roots. But in order to make you understand more on that mechanism, it is better to use the model which has been constructed by a scientist known as Much in 1930. This scientist constructed the following model. Okay. The model looks like that diagram. The diagram here, this one is considered to be A. A is filled with the solution which is high concentrated. And this one is considered to be C. C also is filled with the solution, but that solution contains low concentration. Low concentration dissolved salts. At the same time, A and C have been connected by a tube, this tube. And here in this container, the container have been filled with the water. So we call this one as D. And initially we expect osmosis to take place in A and C, meaning that more water will enter to A, at the same time water will enter to C. But because this A contains high solute concentration, more water will enter to A rather than C. The entry of water here in A creates hydrostatic pressure in this ABC. create pressure in A, B, C. A is this one, B, this tube, C, this one. So hydrostatic pressure here is created. The hydrostatic pressure which is created there makes materials which are found in A here to move or to be transported from A to C through B. And when your materials are moved from A, it means the concentration here becomes lower. That phenomenon will continue until equilibrium is reached. When the concentration in A and C becomes the same, then that phenomenon stops. But mind you that this is a physical model as explained by March. This model can also be seen here in this diagram, which is kept here. It is similar to that one. You have A, C together with the B. B have the solute concentration here is more negative. It means it is concentrated compared to C. This creates hydrostatic pressure which causes water and minerals to move now from A to C. You see? And this is D. D is composed of water. So this is according to March's hypothesis. Therefore, we can compare that model with the living plants. For the case of plants, they work similar to the model which we have seen there. But equilibrium is not reached. Equilibrium is not reached because we expect food materials in A there acts as leaves, photosynthetic cells. They synthesize food. So leaves will have high solute concentration. And B is considered to be the phloem through which food can pass through. C is considered to be the sink. Sink like roots and all other parts of the plant which need food. Leaves here are considered to be the source. The source produces food. That food goes to the sink. They dump to the sink. The equilibrium is not reached here because sink uses that food. The food is continuously used. At the same time, food is continuously produced. And D here, D acts as the xylem for conducting water. But as I have said earlier, the model is too physical. We don't expect equilibrium to be reached in living cells. Also, the model does not explain about why the phloem should be living and active. The model failed to explain on that. Okay. Then, as usual, if we want to understand more about how that model works and how that model can be applied in living plants, you are required to sit down and try to answer that question. You are required to explain it in details. After explaining it, you are required to mark your work. Marking is not necessarily to be done by teacher. Even yourself, you can mark your work. How? You compare what has been written in the books and what you have written there. If you find out that you have mixed things there, you read a book, you understand each and everything from there, then you try to answer that question again. If you master to answer that question, then you can assess yourself that you have achieved that goal. I think you have understood. So please, I beg you, answer all the questions which have been presented here. Mind you that I gave you just the skeletons in each question. You are required to put muscles in those skeletons which have been given by me. If you put muscles, then obviously the goals will be achieved. And up to here, I'm through with my presentation here. We have started from several things. We made about 3 presentations, I think you remember. We started with the vascular tissues, their descriptions, adaptations. We went to the transportation of minerals and the water. Today we concentrated on transpiration together with the transportation of food. It is my hope that you will cover other parts concerning with the transportation in plants. Next time, I will come and present to you on transportation in animals. Try to check various past papers based on that topic. Check the question there and answer the questions which are found in past papers. When you do several questions, I hope that you will be in a better position to understand each and everything concerning with the topic of transportation. Thank you very much.